I'm Giulia Babbi, working at the Biocomputing Group of the University of Bologna, and I will describe eDGAR+, a data resource of annotated gene-variant disease relations. In order to overcome the problem of retrieving the comprehensive annotation of genes associated with diseases, we implemented eDGAR, an online resource of gene-disease associations with a specific focus on the annotated relations among genes involved in the same disease. Here we present the upgraded version of eDGAR, eDGAR+. In Table 1, we show the statistics on the new database, including data from MunsaVar, CleanVar, OMIM, and DisGenet, for a total of about 17,000 associations among 8,000 genes and more than 5,800 diseases. In Table 2, we show the incremental content of eDGAR+ with respect to eDGAR previous version. Considering unique gene disease association, removing redundancy among the databases, we obtain figures that are much higher than the previous version of eDGAR. Gene disease association from created dataset in eDGAR+ are triplicated. We revised our annotation pipeline, Figure 1, supplementing each gene disease association with the available information on functional annotation in terms of gene ontology, Go terms, KEGG and reactome pathways, and EC numbers. Structural and functional characterization of proteins as inferred with the Bologna Annotation Resource 3.0. Characterization of diseases in terms of disease ontology and human phenotype ontology. Known variations and their relation with diseases, expression of proteins in different tissues and cell types as retrieved from the Human Protein Atlas, transcription factors involved in protein expression and regulation, the subcellular localization as predicted with BUSCA, protein-protein interactions in transient and stem complexes, interaction of proteins with small molecules and drugs as reported in Campbell and Drug Bank. We adopted the newly released NetGet Plus as enrichment procedure to endow the sets of genes, proteins involved in the same disease with characterization in terms of biological processes and pathways possibly involved in the disease etiology, including Go, KEGG, and reactome terms. Concluding, eDGAR+ is a curated and comprehensive resource for retrieving data on gene disease associations and related annotations. It updates the previous version, eDGAR, under several aspects, since it includes new data from different primary resources, new features, and sources of gene annotation. We present eDGAR+ as a resource to help researchers in directing their analysis, in particular for highlighting the features shared by variants, genes, and proteins associated with the same or similar diseases. eDGAR+ will be soon available online with an improved web interface that is under preparation. We invite you to visit our website and we thank you for your attention.